matrices.
Oh,
Yeah, I looked at it, I think that's a matrix. I don't know what that is anymore, but I remember having to do it.
The speaker is the room speaker, it's not this thing.
So he said, yeah, I don't know what. I said, well, I don't know what that is.
Try the real
I know the statistic is awesome throughout your life, because so many things depend on statistics.
display audio.
Impressions and
Try
all
display
that
audio.
stuff. I mean, I never used calculus in my whole life, but statistics, absolutely. And so it's like, yeah, that's the really useful math, and you got the right guy to have it.
Maybe talk.
Eric knows his-
Can you try talking to us again, Jeff?
Yeah, how's this sound?
That sounds way better because we can hear you.
Awesome. Okay, you can see the screen okay?
Yep.
Okay, great.
I don't think he knows his age. He's a freshman. He was a red shirt on the. I don't know.
Fed in through the, yeah.
Elk shed collector. Elk hunting, with a wandering elk shed collector. That's what he was do all the time. Yeah, I was just walking through the basement.
The cookie limits there.
Headlines about him, lives in his mom's basement. Collects sheds for a living. Yeah.
Are you all squared away?
You know.
You all good?
Just has a whole bunch of like really doofy things. Headlines for like on the cover of mediocre.
I'm a bit bummed out.
In fact, they talked about.
I wanted to ask.
And then we went and set up like a year fitting the guy. I'm having a really hard time. In the springtime, just.
You have
And then the first. Sports. Yeah, you do make money sports. Yeah,
Even
savvy.
figure all that stuff out.
Are you all set for your dinner, Jeff? Good.
What these two teams are total, or who will win by what spread, or. Unless you got a surround stand beside you.
Okay. Jeff, can you exercise your voice again? We're checking another system here.
Okay, yeah. You hear me okay now?
Yep.
We'll check when it's quieter.
Okay.
So I'm getting all the background noises.
Right. Okay.
Oh, wow. I'm supposed to shoot, have like several dents all piled up in there, so it's like. Yeah, wow. No way you can figure stuff out.
Oh, yeah.
Every time I go hunting, I think it's shit.
I realize what might be wrong. Have him say something again. I'll say a few seconds.
Okay. Okay, Jeff, want to try again?
Yeah, is this any better? It's all good?
Yep, that worked perfectly.
All right, great.
Thank you.
Just in time, right?
That's right. We have like 3 sound systems trying to tune in here, so. And I'm not that good at technology. Remember the iMac in the iMacs we used in your lab? I think we're ready to go, Jeff. Are you good to go? Okay. All right, everybody. Well, I think we're going to go ahead and get started here. Jeff has a dinner he has to take a colleague out to after this, and it's 2 hours later in Pittsburgh. So we'll get him up and running. So thank you all for coming to the biology seminar series, Life Inside and Out. Thank you, Bradley, for organizing this series for us this semester. So it's my distinct pleasure today to introduce my graduate school advisor from many, many years ago. I was Jeff's second graduate student to go through his lab when he was just starting off as a faculty member at the University of Pittsburgh. So Jeff actually got his Ph.D. at Harvard, and he worked on a project that was involving sodium pumps in the brain. And the common theme that he started to establish at that stage of his career was the idea that the proteins that he was studying assembled themselves in the endoplasmic reticulum, which is an organelle found in eukaryotic cells. And so from there, Jeff transitioned, as he said, to get away from all the blood involved with doing work in brains, and changed instead to yeast, which don't have brains, but do have endoplasmic reticulum. And Jeff went on to do a postdoc in Randy Sheckman's lab at the University of California, Berkeley. Randy since has recently gone on and won the Nobel Prize in Medicine for a lot of the preliminary work that he did on protein transport in eukaryotic cells. And the endoplasmic reticulum is deeply involved in that process. So Jeff did a lot of work in Randy's lab, working on molecular chaperones. There's about 10 or 11 of my students out here, Jeff, and I talked to them about molecular chaperones yesterday, so they should know all about that. And so Jeff is focused on protein movement within eukaryotic cells. Also in protein quality control is what he's been more recently interested in. Proteins have to fold properly. If they don't fold properly, bad things can happen if the cell doesn't deal with it. Two ways of dealing with it: you can retry to fold again, or you can get rid of them. And so what Jeff's going to talk to us about today is, well, those mechanisms of ensuring how cells get rid of trash. So I'll just add before I shut up here, I'm a professor, I like to talk. Jeff was one of the most fabulous mentors and advisors that I could have ever dreamed up or drawn out for myself. And I was just lucky to be able to stumble into this young professor's lab early on in his career. Jeff was extremely supportive of my career choices, which was to do something like this, teach at a small school. And Jeff encouraged me and supported me to go and get extra teaching experience to make me better at that job. And he also taught me how to be a great mentor myself and do research with students like many of you undergrads. So thank you, Jeff, and thank you especially for coming and joining us via Zoom here in Montana. So
great. Thank you so much, Mike. You know, it's easy to be a great mentor when you have great students. So, you know, I owe it to people like you. So thank you. Yeah, no, thank you for having me present to you today. I really want to come visit in person someday. I love the West, and you know, it's in my future, I know for a fact. So hopefully I'll meet you in person someday. So thank you so much. This is Pittsburgh. The next thing is for Mike, because I think his favorite thing in Pittsburgh is actually shown here. Mike is a huge hockey fan, as you probably know, and Penguins are not having such a good year this year, but Mike, come back and we'll go see a game together, and that'll do it.
that sounds perfect.
All right, excellent. Yes, they need some help. So I'm going to start by talking about this. And, you know, somewhere back in a textbook, you opened a page or heard a talk, and there are all these like colorful things, right? And these are, you know, the diverse proteins that exist in nature. And there's ways to predict their shapes. It's probably going to be a future Nobel Prize. But, you know, the colors are added, of course. Proteins don't necessarily have color, some do. But, you know, proteins are made up of beta sheets and alpha helices. Sometimes they form these really cool beta barrel structures. This is one of these like helical type beta barrel structures. You have these short beta sheets and lots of alpha helix. Proteins can come together into complexes. In fact, most of the proteins that you make inside your cell don't exist alone, but they exist in large complexes. And I'm sure a lot of you have been, learning about the Krebs cycle, glycolysis, and all this stuff. And these things are always taught as here's an arrow, here's a reaction. Here's an arrow, here's a reaction. But actually in the cell, all those things clump together into one large complex, which makes things happen more efficiently. So that's a pretty complex decision, right, itself. You have to fold these things into these beautiful shapes. They have to find their partners. And as probably some of you know, they get these modifications. Some may have lipids attached to them or phosphate. They form disulfide bonds. Cysteines have to form disulfide bonds. Sugars are put on most proteins that are secreted. So there's a lot of things happen in this assembly line. Like any assembly line, at the end of the assembly line, you have to have someone say, this is good or this is bad, right? So this is a good example here of what happens if the quality control manager isn't doing their job. These are various model systems. These are worms that are expressing the protein that's linked to Huntington's disease. And you can see that if you increase the repeats that form aggregation propensity, the protein, instead of being soluble, becomes these pumped up. And that's what sadly, kills people with Huntington's disease. One thing we've worked on for many years is a disease called antitrypsin deficiency. And these are giant globules of protein that form inside liver cells. Eventually, these liver cells will die and people get cirrhosis. And these kids, if they don't have a liver transplant in a year or so, will die. Cystic fibrosis is very common, and actually that's going to be one of the vignettes I'm going to tell you about later. And sometimes the protein linked to cystic fibrosis can form these large inclusions. And then Lewy bodies, which exist in Parkinson's disease, are again these sort of large aggregates that form. And since some of you just heard Mike talk about chaperones, chaperones should be doing the job to prevent all these bad things from occurring. They should keep the protein soluble. But over time, as you age especially, those misfolded proteins just start accumulating unless there's a way to destroy them, right? And even if you're destroying the bad stuff, sometimes it still accumulates. So there's sort of this battle between being a good quality control manager and simply age as misfolded proteins accumulate. Now, some of these happen spontaneously, and a lot happen because of mutations, so genetic mutations that predispose us all to diseases. I'm going to say a bit more about this in a second. All right, so it's difficult to fold proteins. I gave you an example. And if you sort of remember chemistry, there's these free energy diagrams, and when proteins are unfolded, they start off at very high energy. And as they fold, they go down these various wells, these folding intermediates, where they start to look like a protein. And if they're lucky, they get to this native state. And chaperones are helping them get to that native state. Chaperones are also, in principle, helping them get to these bad things, these aggregates. And in fact, the most stable type of protein that exists are these amyloid fibers that you see in, for example, Alzheimer's disease. And the reason they're so stable is because they're lower energy than the native state. So we want to try to prevent that. So chaperones should be doing their job, and maybe degrading these intermediates that aren't going to fold is a good thing to do. And then here's the scary news. 1 out of 50 people carries a genetic mutation for a catastrophic disease, right? Now, the odds of course, you finding another individual, there's many catastrophic diseases, the odds of you finding that other individual having kids, 1 in 4 chance, right? The odds are not high, but they're substantial enough that this is a major burden on the healthcare society, healthcare in general. And once vaccines get better and COVID is less of an issue, these problems are still going to exist, right? So where might protein folding in the cell be most problematic? So you've seen pictures like this before, in a eukaryotic cell, there's the workhorse of the cell, the mitochondria, which everyone says is the powerhouse. It makes ATP. Although not drawn here to true scale, the endoplasmic reticulum takes up the space of almost the entire cell. It's usually shown as just kind of hugging the nucleus, but it's everywhere. There's ribosomes that pop onto the endoplasmic reticulum. There's Golgi apparatus that acts as a way station for secreted proteins that are made in the ER, go to the Golgi, go to the plasma membrane. All those plasma membrane proteins and all those things that your cells secrete have to go through the secretory pathway. Roxosomes, not shown here is the lysosome, which actually is an important organelle for storage. And, you know, in truth, the place where the protein folding problem is the biggest deal is the endoplasmic reticulum. Here's an example of a yeast cell, a thin section, electron microscope thin section. And you can see these beautiful strands of ER. I apologize, this is actually a liver cell because liver cells make and secrete a lot of proteins, so they're just loaded with ER. But all this space in the cell is basically ER. And it's hard to see here, but they're all studded with little ribosomes, chugging out lots of protein. And I think the stat, which is amazing, is that a liver cell will secrete its own weight in protein like every week. So they're huge protein factories. So that's pretty stressful on these little threads of endoplasmic reticulum. Here's yeast, right? And here it's not quite as profound. Yeast don't secrete so much protein, but you know, it's still pretty easy to see this strand of ER that's going to take care of a lot of protein. So why should we care about the endoplasmic reticulum in particular, this first step in the secreted pathway, a place where a lot of proteins are made? Well, as I said, 1/3 actually of all proteins enter the endoplasmic reticulum, 1/3. You and I make over 20, 30, maybe 40,000 different proteins. That means 1/3 of those proteins right now in your body are entering the strand of endoplasmic reticulum. So somewhere between 8,000 and 10,000 proteins right now are entering your ER. They have to be folded. They have to become modified with all those things I talked about, sugars, lipids, phosphate. They have to find their partners. On top of which, the ER is where you synthesize fats and cholesterol. And you know, we've all seen commercials, and some of us are taking statins to reduce cholesterol. Well, that is attacking an enzyme in the ER that makes lipids, it makes cholesterol, and that's how statins work, right? And not surprisingly, many human diseases are related to problems with protein folding in the ER and fat metabolism. And in fact, a quarter of the class that I'm looking at right now will die from coronary artery disease, which is basically linked to misregulated fat metabolism in the endoplasmic reticulum. This is such a happy day. Okay, I'm trying to waste your time. You're at my people I'm lecturing to. All right, so back to the secretory pathway in a cartoon form. So here's a cartoon version of the endoplasmic reticulum, and in this beautiful picture from a classic textbook, you know, there's red proteins and little blue proteins and membrane proteins, and everyone knows where to go, and they all go through the Golgi happily. And there's vesicles that take blue ones and red ones, right? It all just seems like such a well-oiled machine. And this is all false, right? Because so many mistakes happen during this process because of the complexity that I've been talking about. So when I started my lab, or soon after actually, when Dr. Morrow was still Mr. Morrow, we became interested in trying to think about how misfolded proteins are handled in the ER. And Mike's work was actually just understanding how ribosomes attach to the ER to chug those proteins into the ER. So, you know, Mike was working on this critical upstream pathway. And more, the literature was sort of suggesting that after, you know, the stuff that Mike worked on, you have these proteins in the ER, and then bad things sometimes happen. So how are they handled? So there are all these hints in the literature that the ER might be the place, the ER might be the place. It could be there. It might be there, but this thing, the proteasome's not important. There's maybe some cysteine proteases in the ER. So the idea was that within the ER, you had this little Pac-Man protease that swam around and found bad proteins and destroyed them. That was the limit of our knowledge in 1995, 1996, which isn't that long ago, actually, in science time. So we did something that was maybe a little stupid, but we named the pathway before knowing really what it was. And it was called ER-associated degradation, or ERAD, and that name has stuck. And to sort of cut to the chase, there's now 70 or so human diseases linked to the ERAD pathway. And the thing that we wanted to set out to answer was, how are these misfolded proteins that are targeted for degradation, ER-associated degradation of bad proteins, how are they selected? How do you choose good from bad? Are these molecular chaperones required since they help things fold? Maybe they help things get degraded. And more importantly, what's the Pac-Man? What's the protease that destroys these things? So we did something that actually Mike knew how to do very well. And Mike, if I remember, you had this undergraduate, Stephanie, who made lots of microsomes for you. Right?
Yep.
And so we take these vesicles from the ER called microsomes. And these ER microsomes are really cool because you can make like a mini ER in a test tube. And what you can do in those test tubes is you can add a wild type protein or a protein that has some folding defect. And it's not very hard to actually get them into these little vesicles in the test tube. And these little parts that were cut off are called signal sequences, which are sequences that direct proteins to the ER. And then the other amazing thing happens in the ER, which I sort of mentioned, is that a lot of them get sugars put on them. So in these little ER vesicles, we made a wild type protein with sugars. And we made a protein that was mutated so it couldn't get the sugars, so it misfolded. And then the question was, well, where's the Pac-Man, right? Because it should be destroying this misfolded guy and leaving this one alone. But that didn't happen. These proteins are totally stable. And when I say, one thing I'd like to say is if you're a biochemist and you don't know what to do next, you add energy, ATP, and you add lots of cytosol, right? Because this was just in buffer, there's no cytosol out there, there's cytoplasm. So that's what we did. We added cytoplasm and we added ATP. And then, like a miracle, we saw this thing pop out of the ER. So somehow, a decision to get this thing out was made by cytosol, and it probably required energy. And even better, within like minutes, that vanished. All right, so that meant that somehow there was a selection process going on. There wasn't a Pac-Man in the ER after all. Something was happening outside the ER, in the cytoplasm. The hard part now is kicking it out of the ER and then destroying it. Now, the beauty of doing this in like yeast is that there's all these mutants we can use. We didn't have to make cytosol from wild type cells. We can make the cytosol from a mutant cell and ask, oh, does it still get out? Does it still get selected, right? And we can do the same thing with the microsomes. And the bottom line is if we use yeast cytosol that was defective for something called the proteasome, now the substrate came out but didn't get destroyed. And what that told us is that this thing called a proteasome, a protease complex, was involved in destroying it. It didn't really matter so much for getting out, at least this activity, but it was destroying the thing. So then we went for it. We purified proteasomes, and there are these beautiful large structures. There's these caps that select misfolded proteins. There's this core that has all these proteases that destroy proteins. And we added nothing but the proteasome and energy, and we could recapitulate the whole event. So that was pretty cool. It meant that selection was occurring somehow. Things were being pulled out by this proteasome in an energy-dependent manner and then getting destroyed all at once. Now, since these caps are actually ATP requiring, we then said, well, let's just add the cap and ATP. And that was enough to get the thing out. And then we added the core part, which has the protease, and that was enough to destroy it. So basically, we recapitulated a selection process for bad proteins misfolded. We know how it gets out. We know how it gets destroyed. And all it takes is the protease. Now, what's the decision in the ER? To do that, a lot of work went into efforts, even back, Mike, when I was pipetting. And the bottom line is that by using yeast with various mutations, we discovered various chaperones that are required for the selection process. And the main ones fall under two categories called HSP 70s and HSP 40s. I don't have time to tell you all about them because that's an entire lecture, which maybe some of you have heard already. But yeah, these are amazing, decision makers that are able to bind misfolded proteins selectively and do something. And they were doing something with this molecule, this misfolded protein. I think our first hint that disease was going to be relevant is that when we had mutants in some of these chaperones, the substrate, instead of getting out, it aggregated into ER. And I'm sure a lot of you appreciate that those aggregates are bad things, right? So if we were getting aggregation in the ER in the absence of these guys, that's probably going to matter. So after all this, we said, and the field was kind of telling us, you got to think about mammalian proteins because there's probably a lot of diseases that result in this ERAD pathway. And, to sort of summarize a lot of work from a lot of people who suffered in my lab over the last, 25, 26 years, we've recapitulated various diseases using yeast as a model. And in essence, what we do is we use yeast as a biofactory. We engineer the yeast to make a misfolded protein linked to a disease. We ask whether it's targeted for the ERAD pathway. We can use genetics and this assay that I told you about and sort of figure out the factors that are required to select and degrade these disease-causing proteins. Because then we can slow degradation and maybe help them fold better. In principle, you can cure the disease. And that's basically the vignette I'm going to tell you about, among many stories when we get a little further into my talk. So sadly, no shortage of diseases. Like I said, there's now 70 linked to this ERAD pathway. They're all proteins that go to the ER. They misfold. The quality control machinery says, we're targeting you for degradation. And then the proteasome eats them, basically. And they're quite diverse. I mean, we purposely chose substrates to work on that are soluble in the ER, that are still hooked to the ribosome, and the ERAD decision is made. Some of them are multimeric proteins that have to assemble between 3 subunits. Some of them are just big, and I'll talk about this one a little later. Some of them are monomers that then tetramerize based on self-assembly principles. And some of them are just a mess, So the bottom line is that if it's hard to fold proteins to begin with, it probably doesn't take much if there's a genetic mutation to push it over the edge. And what's amazing, the more and more we've done this, here's a protein that you and I are making right now, and you and I are degrading probably half of the protein, even without mutations, because it is so hard to fold. I mean, this cartoon doesn't do justice. They're so hard to fold that your quality control manager says, I can't fold this thing. It's taking too long. It's not getting the right modifications. So you just destroy them. It's very American. You make too much, you throw it away, right? So, this is how cells evolve because, as we like to say, sometimes for a cell, it's better to just trash something quickly than to exert more effort trying to fold it. Take what you get and run with it. And that's what our cells evolve to do. They take the good proteins, anything that's slow, they just, they don't wait around for the protein to fold. And that's the problem with a lot of these diseases is that they may be a little slow to fold, but if given the opportunity, maybe it's not such a problem after all. So there's a lot of big questions we're asking in the lab and that we've been asking over a lot of years. One of them is, can we develop drugs to actually target this pathway, as I just mentioned, to maybe slow this degradation process to allow proteins to fold better? Are there backup systems for ERAD? And I think, for all of you biologists in the room, anytime you learn about how decisions are made, if something goes wrong, there's always a backup system, right? There's always ways to repair DNA damage. There's always ways to refold proteins. There's always ways, if you put the wrong amino acid into a protein in the ribosome, you can edit it out again. This is what evolution does, right? You make this machine better and better and better. And it's selected for, because each time there's something that makes the machine work better, those cells have an advantage. That organism has an advantage. So, a billion years is a long time. So there's got to be backup systems. And in fact, we started finding them. There's some misfolded proteins that escape the ERAD pathway, but they're caught later, which is pretty cool. What does a misfolded protein look like, right? What are the biophysical features that lead to its selection? And that's the hardest problem I think we're working on now, but we have some hints finally. Some of these membrane proteins, right? They're being eaten by the proteasome, which is in solution. So that means by definition, you have a membrane protein in a lipid bilayer in an oily environment. And that membrane protein has to somehow get into the aqueous phase, right? So that's tough. And there's actually chaperones that can hold membrane proteins in solution, which is just crazy. So we're doing more work on that. And finally, a big problem, and I think something we're doing in the future, is there's so much genomic information, sequence analysis. I know like for the neonatal intensive care unit here, you know, it's really hard to treat a baby. They can't tell you how they're feeling. But if they're really ill, you know, with the parents' consent, they just sequence the entire DNA of the baby, right? And then within days, you say, well, here's the genetic mutation. Here's the metabolic disease. So you can treat the kids, and, you know, instead of doing all these horrible invasive things in a newborn. So there's a lot of genetic information out there, and guess what? A lot of these mutations that cause disease are causing misfolding in the endoplasmic reticulum. So again, it all sort of comes back to ERAD. I can't tell you all these stories, but I'm going to tell you a very quick one about this first one. So I think it might be interesting to you. Is there a way to drug this pathway to treat what we call a protein conformation, a protein shape disease? All right, so here's the disease I'm going to tell you about, mainly because in some ways it's been a hard one to work on, but now clinically it's like one of the more treatable ones, which is just, it's such a wonderful case study for why science is a good thing. So cystic fibrosis is the most common fatal inherited disease in Caucasians in the US. When I was young, the life expectancy was about 10. And if you know someone with cystic fibrosis or you've heard about it, you know, basically it's mostly a respiratory problem. And the problem is simply that the mucus in your trachea, in your lungs, the mucus is too thick. And that's because this protein, CFTR, moves chloride out of the lung, out of cells into the lung. And where chloride goes, so goes water. So people with this disease don't properly hydrate the mucus. So they have really thick mucus. And in the old days, kids would actually just like asphyxiate from the mucus. A horrible, horrible death, right? And now it's mostly because of opportunistic infections. The thick mucus makes basically a hospitable environment for really bad pathogens, some of which are just difficult to treat. The good news though now is the life expectancy is like in the 50s. And I'll tell you one of the reasons why in a bit. So from 10 to 50, people are living a lot longer, they're living healthier, and given new drugs, I think that's going to continue to get better and better. So it's really one of the wonder stories where you clone a gene, you have disease population that's really involved in the science, you figure out the cellular defect, and you try to fix it. So the protein, as I mentioned, is CFTR, that's short for cystic fibrosis transmembrane conductance regulator. It's a mouthful, so CFTR. And in 90% of the patients, guess what happens, right? The protein is actually made, but it just can't get to the plasma membrane where it's supposed to be pumping chloride. This is an example that's crazy, right? 80% of the wild type protein, 80% of the protein you're making right now is destroyed. It's just so hard to fold. But the bad news is if you have the disease-causing mutants, 100% disagree, right? And you can probably guess the pathway that degrades it. It's a membrane protein, it's made in the ER, it doesn't get synthesized and fold and get transported into the plasma membrane, so you just kill it, right? It doesn't fold well. And this is a relatively recent structure, and which has really given us a lot of insight, but it's a huge protein. It actually takes 8 minutes to synthesize. You know, you think of biology and microscopic things being fast. No, this takes 8 minutes to synthesize. It takes another half hour for it to fold and then even get out of your ER. Again, this is happening in the best case scenario in all of you. But if you have this mutation, you know, the cell says, wait, there's a mutation. I'm not going to wait around these 8 minutes to make you. I'm not going to wait around 30 minutes to get you to fold. It's just destroyed by the ERAD pathway. So maybe if we gave this protein more time, you could start to fix it. And the first hint that you could fix it goes way back. Yes, Mike, even before I started my lab. And this is a really wild experiment, but I mentioned that proteins get sugars put on them. And if the sugars are put on the ER, it's called this B band. And if the protein goes to the Golgi, it gets more sugars, and that's called the C band. So just by running up a gel and stating it, you can say immature, mature. right? ER, Golgi, and mature. You just look for that. Now, some of the best experiment, and this is the disease, right? It's just stuck in the ER, and that protein is getting degraded, and this is you and I, hopefully, right? We're mostly making this good one, even though it's kind of degraded to some extent, it's mature. Now, probably an overworked graduate student, instead of putting his disease cells at 37 degrees, he threw it in a colder incubator. And guess what he saw? Just lowering the temperature got some of the disease protein to now mature. So this disease is actually a temperature sensitive disease. If you just lower the temperature, in principle, you could cure the disease. You're allowing the protein to fold better and get out of the ER. So you sadly can't cool people's lungs who have the disease, right? But this gave the field the idea that there was a way to fix the folding, because on the chemical side, low temperature favors hydrophobic contacts and slows ERAD, and it favors probably chaperone binding. So, this all sort of made sense. So this is 1992, and now we're going to jump to 2015, and the first FDA approved cocktail came out through a lot of work and a lot of effort. What this cocktail is a 2-drug combination. The first one, created by Vertex, basically takes this defective thing in the ER, and instead of allowing it to get fully degraded by the proteasome through ERAD, fixes the folding partially. So you now have a better looking protein that allows it to get out of the Golgi. It goes to the plasma membrane, and to make it a better chloride channel, you add the second drug. It's not totally fixed. You got to make it a better chloride channel, and now it works. And now water can flow into the lung. People's mucus isn't as thick. That's it, right? This 2-drug combination. Who knows how many millions and billions of dollars it is, but it's saving lives, right? So this is good news, right? Now there's problems, which is that it doesn't completely fix the fold. And the chloride transport's not quite as good as you want it to be. So these people aren't totally cured. There's some side effects too. Some people stop taking the drug because it's a little nasty sometimes. And there's still some residual problems. So the question is, can we do better, right? And there are now other correctors. This is one that came out just a couple of years ago, but now there's a third compound that's added in. And this is a change in how well you're breathing, forced excitatory volume, which is how well you can, blow out air after inhaling. And the bottom line is, if you have the disease, this is where you're at. If you start taking these medicines, you just go up, and then it kind of plateaus nicely. I was at the Cystic Fibrosis National Meeting last month, and this was early data. This is week 4. They now have 3-year data showing that the compounds are still working. So that's awesome, right? And again, it's really good news. It helps fix this protein. It helps fix the disease. It's not perfect. And there's one other caveat: $200,000 a year. So this is a topic for another time, but that's what the drug company charges, and they have a monopoly on this. So there's a lot of interest in finding other ways to do this. You can ask me about that later. All right, so this is what's out there. Can we still do better, right? And one thing I haven't told you about most of these ERAD substrates, like this protein CFTR, is that they get modified with a small peptide called ubiquitin. And ubiquitin is really important to make sure that the proteasome finds you, that protease comes and finds you. And this pathway is pretty complex. The bottom line is it's a 3-enzyme cascade where this little ubiquitin guy is hooked onto a substrate, and you form these chains. And that allows the targeting of the ubiquitinated protein to the protease. All right, so none of the drugs that address this. And it seemed to make sense to us that, hey, you may make the protein fold better, but if you can somehow block this ubiquitin pathway, so now things just don't get recognized as well by the proteasome, maybe you'd save those proteins. Maybe they could have a better chance to hang out and get targeted by the drugs, right? So let's block the tagging. They don't go to the proteasome. There's a bigger pool of protein now available for those drugs to. So we needed to go back and use an assay that we published a long time ago now. But we take human cells that express this protein. We make those little vesicles like Mike was using when he was a graduate student, and I told you about before, right? These ER vesicles. And then we add this ubiquitin molecule with a radioactive tag, and we add cytosol and ATP. And what's really cool is we can actually, in this assay, watch the radioactive ubiquitin get attached to the misfolded protein in these vesicles just by precipitating it. And I don't think I have a gel to show you, but the bottom line, actually, there's a gel, and what you're looking at is this ubiquitin schmear, because the ubiquitin chains can be various lengths. There's the radioactive ubiquitin smear. But now if we add this at low concentrations, if we add this inhibitor, ubiquitination, you see it kind of goes away. So hey, maybe now the protein that doesn't have this tag, the CFTR protein that doesn't have this tag, will be able to sit around longer and then get fixed. And indeed, that's what we saw. So this is actually a pretty intricate assay. You're just gonna have to believe me. But this is measuring current, ion transport, chloride transport across lung epithelial cells. We did this in collaboration with a colleague. And if you add this is a shorthand for that FDA approved compound. Look, you do a little better, yay. And if we add our compound that slows that ubiquitination, you know, it's kind of statistically significant. But if you add these two together, you get this nice synergistic response. So this plus this is not as big as this. So by slowing this ubiquitination, by slowing ERAD, slowing degradation, and now fixing the protein with a folding thing, you know, make a better channel, right? And there's more of it, which is awesome. Now, this fancy compound that we used here is really kind of toxic. I hinted at that because we couldn't use too much of it in these assays. But Jen, who Mike actually knows, still in my lab, and we had her involved in this project. And we started collaborating with Pfizer, who had a real interest in this pathway. And actually at that time I was consulting at Pfizer. I was consulted there for like 7 years on their cystic fibrosis project. And I got them kind of excited by this. So what they did was they looked at the structure of this one toxic compound, and they helped us select and make all these other derivatives. And this is essentially how well those derivatives work. They're all slight structural variations. So if you're taking OChem, they actually say, oh yeah, this is kind of like this one, this is different, and this is different, you know, all these features. And we also, what was good was we found some that weren't so toxic. This is a GI50, which is a measurement basically how toxic they are. So in fact, we have this really potent one, 7134, that is more potent than the parent compound. It inhibited that ubiquitination nicely, but it also wasn't toxic. And what we later published was good. So here's now that kind of assay. This is actually a different assay. Here we're actually measuring the ability of the protein to get to the plasma membrane. And, you know, we also did functional assays. But this is a really cool assay because you can actually say, is the protein getting out of the ER and at the plasma membrane? Basically, we put a fluorescent tag on the protein. With our corrector, this is what you get. With the FDA-approved compounds, this is what you get. And if you mix them together, you do that. And the cells are fine. Working with a computational chemist, he also modeled the binding site for our compound within this ubiquitin enzyme. So this is hope, right? This is sort of that first step toward now starting to really think about trying to make a drug. So that's one good piece of news, right? Is for this disease, cystic fibrosis, again, there's very good FDA-approved drugs out there. They're not fixing everything, but the goal, of course, is that to try to make those existing drugs better. The bad news is there's all these other proteins out there that are misfolded, being targeted for degradation. If drugs could come out that make them fold better, I think we can start to think about ways that we can now address that problem, right? By inhibiting the ubiquitin pathway to make more protein available for those drugs to work. As I mentioned before, 70 diseases linked to this pathway, and it means there's no shortage of work in the future. So everything ends up being a big team. I think here's the current lab, a terrific team. Again, Jen did some of the key work here. Chris Guerrero did a lot of the initial work. Past students and a postdoc also worked on these projects. I have a lot of collaborators for all those different substrates we work on, many of whom are MDs actually, but at least for the project I just told you about, besides Pfizer, Peter Whip, who's a colleague at the University of Pittsburgh, we worked with for a long time. Lots of funding, it takes money to do all this stuff. And finally, it takes these awesome people. And I have no idea who these two people are, Mike, but
those are young people for sure.
Who are those young people? Yeah. I didn't have a beard, I didn't have gray hair. But anyway, thank you so much. And I'm happy to take any questions, no questions too naive. So please ask away.
Awesome. Thank you, Jeff. Questions? So Jeff, have you guys done, use the ubiquitination inhibitors on any of these other disease proteins other than CFTR? Have you done those experiments or?
Yeah, that's an excellent question. Excuse me. So what we know is that you have to sort of use them where there's already hints of something that's going to make the protein fold better.
Gotcha.
Right?
Mm-hmm.
And one thing we've done instead is, because there aren't really drugs for all of those other diseases, those 69 other ones, right? There's some being developed, and we've started working with some of those folks, but actually we can't do it all, so other folks are doing it. We're kind of sometimes just advising them. Yeah. But the one thing we can do is we can just take those cells making these misfolded proteins and just lower the temperature, right? Mm-hmm. And then if it fixes the thing, then we can kind of do these cool synergistic studies where you sort of lower the temperature a little bit, now you add the drug, right? See if it does better. So it tells you you're on the right track. That means that eventually when there are drugs, maybe they'll have a chance of working.
Yeah.
So yeah, it's a tough one. The field is really interested in this, and there's another disease, antitrypsin deficiency, which is one of those many substrates I showed. And yeah, there's a lot of work trying to make some what I call chemical chaperones to make that fold better. Chemical chaperones are pretty cool. They're not so specific. They don't just recognize one substrate. They basically generally make the ER a happier place. And these chemical chaperones just improve the ER environment. And those kind of work on a lot of these. So we're testing one now. We actually have a mouse model now in the lab where there's a lot of human cell work too, but yeah, we have a new mouse model for a protein linked to kidney disease, and we're injecting mice with the chemical chaperone. And that does the job, which it hints maybe it's working, then we can start to do these combination studies. Yeah, it's a brave new world. Right?
Yeah, it's awesome.
Yeah, it's fun.
Anybody else? Yeah,
Anyone
Jack,
else?
Jack.
I once upon a time used to show an old film in this lab when I had respiratory system to talk about, and it was one of these, videos about a young lady in Great Britain, a girl that had CF, and maybe you saw this old film back in the day, and the whole focus at that time was that, I think, as I recall, longer chains of DNA were being, they were breathing in stuff to kind of chop those smaller so it'd be easier for her to do the shifting of material and coughing up this thicker, I don't know if you ever saw that, but they were just looking at how do we take care of the symptoms, not what you're doing, which is to get to the root of the problem. But how do these things that are moving fluids in, how does that relate to these longer pieces of DNA that they're trying to just chop?
Yeah, that's an awesome comment and question, because I sort of said, the life expectancy was 10 and now it's like zoomed up. A lot of that zooming up was even before these drugs came out in 2015. It's because of stuff like what you just said, right? So in that mucousy environment, it's not just the mucus, which is secreted from the cells, but you have a lot of long DNA shreds, which sometimes come from pathogens. It also comes from just cells lysing and dumping all their nucleic acid into this environment, and that clogs it even more. So yeah, it's basically an inhalable DNase. People inhale the enzyme that just chops the DNA up, right? So it kind of helps it become a little more, movable. And then people often wear these, it's like, it's a vest which you plug in and it just shakes, right? So people with CF wear these for a couple hours a day while they're inhaling this stuff. High salty mist also helps move the mucus a little better. So a lot of this, before there were good drugs, you'd inhale DNase into your lungs. You'd have just movement, so the mucus would move a little better, and they'd cough it up better. But this is just not a way to live, right? And then still get these infections. So that's still done. Even with these drugs, people still do that. And that tells you that the problem's not totally fixed, right? So thank you for that question, because it really ties in beautifully to why things are better, but they're not perfect.
And the other thing I remember from that film is you said this is an environment because of that thick goopy for a lot of different nasty things to develop. So a good incubator. So she was constantly having to have sputum and so forth sampled in which antibiotics. And again, through the misting, taking in a certain numbers of the right kind of chemicals to combat whatever it is now that's in her lungs. I guess that's probably a never-ending problem too.
Never-ending problem, exactly. The major one is Pseudomonas, but there's these mycobacteria. I mean, there's just lots of really nasty stuff. Actually, I have a colleague, Mike, you may remember Graham Hatchell, but he works on mycobacteria, which is one of the pathogens, and he uses bacteriophage. Right? There are viruses that attack bacteria. So he has engineered bacteriophages, phage, for people with CF to inhale. So it starts to eat, they basically invade the mycobacteria, the invading force, right? And it kills them. And it's a tough project, but he's had some amazing case studies. There's some real success stories, but it's tough, right? You got to do it on each patient. And you got to make sure it's clean phage. But that's one way they're trying to attack the bacterial infections, because as probably a lot of it's a war out there, right? The pathogens are going to evolve faster than we can make new drugs. We'll never win that battle. I see Mike writing, which means that I see an exam question in the future.
I just remembered something. I got to write those things down now.
Yeah, I understand. Very much. I do understand.
I have a question. Solid page or comment. Solid page. You have to find a particular, a specific page for specific bacteria.
Yes.
Yeah. Mm-hmm.
So that means you have to propagate those pathogenic bacteria.
Yeah.
You have to, and what they do is they actually get the pathogenic bacteria from the patient.
Yeah.
So they have a specific isotype. Then they go to their -80, which has like 100 different phages, and they laboriously go through and say, okay, number 20, number 15, and number 83 work. They then mix those three together. First, they purify and scale up. They mix those three together. They ship them off to the hospital. There's 3 months of paperwork, and if it works, it works. So there's hits and misses, but it has really saved people's lives. Not many, but so it's small scale stuff. But
who's the researcher that's working on phages?
His name is Graham Hatchell. Mike can tell you all about it. No, he works down the hall from us, and
one of his old students works at Montana Tech in Butte, Montana, just an hour north of here. Marissa, you remember Marissa?
Oh, you're right. Yeah, she was like a Olympian in judo.
Yeah, she was Olympic judo. And graduate.
And an amazing phage researcher.
Yeah, right.
Yeah, she's awesome, right? Because she does the phage project.
Yeah.
Okay, thank you.
So that's cool stuff.
What I was writing down, Jeff, is Jack's question about the old video reminded me that your introduction to me of the Paul Berg translation dance video, it shows up in all of my advanced cell molecular biology classes. So
Excellent. YouTube, yeah, has everything. It's great.
you guys remember that, right? They won't ever forget it. Yeah.
Yes.
Other questions for Dr. Brodsky?
Not, you can always email me. Oh, yeah.
So I'm guessing, I think I'm interpreting this right, that,
Sorry, it's hard to hear you. Sorry.
Here, let me turn the microphone.
Okay, sorry.
so these drugs must have to be pretty substrate specific, right? Because you don't want to mess with the overall degradation process, because
Right.
it sounds like there's a lot that needs to be degraded normally. Am I reading that right?
Great question. So the ones we've been using they're not completely broad-based, but they don't have enough specificity to be in the clinic, right? And that's why, as I said, it's the first step toward that next step. So to maybe get a little more fine-tuned, that ubiquitin pathway, there's E1, E2, E3 enzymes. The E3 is the business end. That's the one that says substrate, ubiquitin, there. These other two enzymes just kind of charge things up. So the E3s are closest to like recognizing substrates. There's still a lot of some diversity, but from a lot of work, including our lab and other labs, we know that there's like 4 of these E3 enzymes that target CFTR. So in principle, if you target those specifically, you'll not completely limit side effects, but you'll do a whole lot better than any other attack. So right now we worked even with another company for a little while on this. So there's a lot of interest in trying to find these E3 specific inhibitors, because then, they can use those as cancer that's linked to some of these ubiquitin pathways. There's all these different misfolding diseases linked to different subsets. The thing that's amazing is you and I have 600 of these E3 ubiquitin ligases. So yeah, and that's because it has to cover the whole proteome, right? It has to cover the 20,000 proteins you make. So those 600, they each like have an assignment sheet, right? It's like getting a homework assignment sheet. So each one has, okay, I'm in charge of these, and I'm in charge of these, and there's a lot. It's a Venn diagram. There's some overlap. There's some distinctions. Some are in with the ER. Some are in the nucleus. They're all over the place. Yeah, that's the next phase of all this stuff is drug discovery targeting these specific E3 ligands. Yeah, it's a great question.
Oh, yeah, there's one more for you, Jeff.
Great.
I was just wondering, sort of computationally, how does the body recognize a misfolded protein, and how does that work? I mean, how do you program it so that you can recognize all of misfolded proteins and not go off and start degrading properly folded proteins?
Yeah, that's an excellent question, right? We had to somehow evolve a system that would recognize 20,000 proteins, with each of them having hundreds of thousands of different conformations and finding the wrong ones. How does that work, right? And the answer at the end is amazingly simple in some ways, but if you have a protein that opens up and misfolds, by definition, what it does is it exposes hydrophobic patches. And if you're a membrane protein, in the ER bilayer, they should never expose hydrophilic patches, right? So if that happens, then something's wrong. So there are chaperones that are not that substrate specific, right? They just find hydrophobic patches, and they find them, and they go through an ATP dependent cycle, and they come up. They find, they release, they find, they release, right? Now stochastically, when they're bound, along comes one of these ubiquitin ligases, and they have specificity for some of the chaperones. So you have a 3-body problem: misfolded protein, chaperones on because something's wrong, and the E3 floats around and says, gotcha. and then adds the ubiquitin to target the protein for degradation. That's an extremely simplified version, but that's one way we do it. There are some of these ubiquitin ligases that are substrate specific, which is incredible. There's also some chaperones that are substrate specific. Some of these proteins are so difficult to make, so expensive, and so important that you evolve to basically have a dedicated chaperone that says, I'm going to help you out, right? Your parents taking you on your first date, watching what you're doing, yeah? You don't want that. It's more like the high school dance where you have the principal out in the corner, sneaking a drink and pretending that he's watching it. So, yeah, that's how our cells work. It's the principle. And he takes a drink sometimes, right? And that's why misfolded proteins accumulate. I've never used that analogy before, and I don't think I ever will again.
Okay, we got one more here for you, Jeff.
Excellent.
Do you believe that with this kind of study that you're doing could eventually lead to making breakthroughs in cancer?
Yeah, I haven't told you about the cancer project we're doing, but we recently published a paper. I had this amazing postdoc in the lab who actually has a job with Pfizer coming up soon. So she's sadly leaving the lab, but much greener pastures. Emphasis on green. She basically screened lots of breast cancer cells for what's their ERAD efficiency like? What's their efficiency of these other degradation pathways? What's their efficiency for chaperones? And it was kind of this really cool compendium. And we went in with these specific hypotheses about, well, triple negative breast cancer cells will be like this, and HER+ will be like this. It was all wrong. Basically, the short story is that some of the cancer lines have very high levels of this quality control manager and the system to degrade misfolded proteins. And they are super resistant to a lot of the chemotherapies that one uses. So, because they have ways to, because what chemotherapies do is they make stress. They make stress on the cancer cells. Cancer cells are already stressed. Chemotherapeutics just make it worse. So if you already have a way to I don't care about the extra stress, right? I have ways to degrade the bad proteins and keep growing. And, cancer cells are a mess. DNA is a mess. They're aneuploidy. They grow fast. They oxidize, lots of reactive oxygen species. So they make a lot of misfolded proteins. So in a tumor, right? A tumor is a clonal selection. So it's a jackpot mutation. One cancer cell has an extra mutation to make its degradative pathways better. And that allows it to outgrow the others. So we're not the only ones to see this, but yeah, I think this whole quality control, it's called proteostasis now, protein homeostasis, but proteostasis and cancer, if you look up those two words and see a lot of papers popping up. That's another 7.
Well, Jeff, we should probably let you go so you can go out to eat with Dr. Rappaport. But thank you so much for taking the time out of your busy schedule to chat with us here in little old Dillon, Montana.
So, this is great. No, thank you for those great questions and your participation and attention, and thank you, Mike.
Yes.
Do this.
Well, thank you. It's been a privilege. We'll see you soon.
Okay. You're welcome. Thank you. Bye-bye.
Thanks for coming, everybody.